Well, the overall impact of genetically modified crops has been extremely positive for the environment. The main thing that genetically modified crops have done to date is reduce the environmental footprint of agriculture. That is to say, reduce pesticide use, where it has that effect from 5 to 70% reduction in pesticide use. But most importantly, it's reduced the use of persistent pesticides that are often a problem for environmental pollution and for human health. But probably the most important thing for genetically modified crops at present is that the crops that are Roundup ready around the world have greatly reduced the use of tillage as a weed control tactic in crops. So as it long been the practice for more than 30 years in southern Australia, where you can spray a paddock, kill the weeds, and go back through and put the seeds in immediately with Roundup. Now you can go across the fields after the crop starts to come up and spray and knock the weeds down, kill the weeds, without using a plow to try to control the weeds. And the net impact of this is greatly to reduce soil erosion by wind and water, and importantly from the standpoint of climate change, because the soil is not being turned over, there's much less carbon in the form of carbon dioxide lost from the soil. The soil has carbon exposed to the air, it volatilizes off as CO2. And that's been estimated at something like 9 billion kilograms per year, less CO2 going in the atmosphere, because of the reduced tillage that's associated with Roundup-ready crops. Well, the way it used to be done was by using a bacterium called agrobacterium that causes crown gall in some plants. And so this is a bacterium that naturally evolved mechanisms to be a genetic engineer. It had plasmids that would insert into plants. But it really only worked for dicotyledonous plants and not terribly well for all of those. After that, John Sanford, whom I knew while I was at Cornell University, invented the so-called gene gun, where he came up with a really, at the time, seemed to all of us to be an absolutely wacky idea of taking a .22 rifle that he'd cut off, basically shaking the powder out of it and just using the primer to launch small gold beads that were coated with DNA into undifferentiated callus tissue. We all thought this was completely crazy. It couldn't possibly work because of course, when we took genetics, position effects of where genes landed was thought to be extremely important for gene expression, where the genes just couldn't express at all. And much to our astonishment, John Sanford actually made this thing work. And so, in many ways, the preferred method for doing transformation of plants now has been the so-called biolytic method. However, the Chinese have actually developed a third method that they popularized. I'm not really sure how effective it is, but they do pollen transformation. That is to say, they transform pollen with the DNA they're interested in, and then use the pollen to pollinate plants and regenerate plants that way. And I'm not altogether familiar with how effective that's been, but the Chinese have made claims that that's how they're doing genetic engineering. Not really, because this is sort of non-Mendelian. That is to say, we're not talking about crossing traits that exist in plants and looking for segregation ratios in the F2 offspring. This is really about introducing genes from other sources into plants. It's much more about transference of genes. I mean, you've asked what's the best way to do it. The best way in some senses is the biolytic method, because it's just, from a technical standpoint, it's so widely available. You can do it in corn and other crops where you really couldn't have done so with agrobacterium. But it's important to remember that as we've done the full genome sequence of more and more plants, we've realized that plants have a lot of exotic genes in them that clearly did not evolve in plants and have been getting moved around by various means, probably most often by insects feeding on one plant and picking up genes, and or viruses that incorporate genes and moving them to other plants. So this technique isn't non-Mendelian, but it is phylogenetically historical. It's transfer among plants. It's been occurring for a long time by one mechanism or another.
Okay.
Well, the classic way they do happens for herbicide resistance is that there will be a mutation in the weed that will make the weed insensitive to a particular herbicide. There are two broad classes of how herbicide resistance can evolve. One is that the plants can have enzyme systems that degrade the herbicide more rapidly, turn it into a water-soluble product and get rid of it. So the pesticides typically are not water-soluble. They're so-called hydrophobic. They attach themselves to oils and lipids and fats and so forth. So metabolism can change them into something that's water soluble and more readily excrete them from the plant or break them down. So one mechanism is by increased metabolism, usually by something called microsomal oxidases. The other method, and that's actually quite common in insects, the other method of evolving resistance to pesticides is by a change in the target site itself. And relatively speaking, this is much more important in plants than it is in insects. So typically what happens is the herbicide would bind to a particular target, and there's a small mutation that's occurred that changes the conformational structure of the protein, so the herbicide doesn't bind very well. And many of the most abundant forms of herbicide resistance in plants are variations off changing the target site.
Okay. And are there any side effects of herbicide resistance on crops? And is this related to the insertion of DNA?
Actually, the insertion of DNA is a really interesting problem. I don't know so much about a specific case in herbicide resistance, but I can tell you about one that happened in cotton. So one of the first genetically transformed crops that was released around the world was insect-resistant cotton. And in the early 1990s, Monsanto released a variety that under laboratory conditions looked to be really good. But when it first started getting field trials in Australia and Mississippi, where I used to work, people noticed that the yields were lower than they were for conventional cotton grown with insecticides. Quite a bit lower, like 25% lower. And this stumped people at the time about what went wrong with this. And it was actually a group at Mississippi State University, where I used to work, had been doing studies on cotton for a long time, and they started measuring various aspects of these cotton plants and found that the bolls in these cotton plants were about 25% smaller than in conventional plants, and that was accounting for the 25% loss of yield. It appears that it was a particular insertional event, that somehow the insect resistance gene, the Bt gene, when inserted, had disrupted something in the plant that caused the bolls not to form as vigorously. All that was really required, and in retrospect it was probably obvious this should have been done, is to go back and look at a wider range of transformants, where you do transformation, you genes insert in various places. So under laboratory conditions, in the greenhouses in St. Louis from Monsanto, the plants looked fine, but when they were tested in the field, this disadvantage was revealed. And now, largely because of that, most people, when they do transformations, instead of putting all their riding on just one transformation event and taking the field, they'll take dozens and do screening in the field until they find the ones that have the optimum characteristics of resistance to insects or resistance to herbicides, as well as good yield characteristics. So you can get these effects, but after that very first case, when people realized, no, you can't produce it that way, look more widely, there really hasn't been a problem. There have been some claims that Roundup resistance seems to confer an increased susceptibility of plants to certain kinds of fungal diseases, but as these have been more scrutinized, there's really no evidence that it's been a strong case. And probably the best evidence of all is, in general, the yields of Roundup-ready crops are much higher than the conventional crops, with or without herbicide protection.
Okay. In what ways does the DNA affect the crops?
No more than conventional crops. Around the world, the safety of GM crops has been intensively studied. The European Union in particular spent millions of dollars looking at possible health risks. And the overwhelming conclusion they've made in reports from 2008 and 2010, and even more recently, is that food from genetically modified crops are at least as safe as any grown from conventional crops. That's a rule we have to use, at least as safe, because we know that conventional foods can often be risky. Peanuts have allergies for people and so on. The main thing is genetically modified crops have been found to be at least as safe as anything produced conventionally. Well, the primary gene that's used for herbicide resistance for Roundup-ready crops, soybeans in particular, the primary gene used around the world is a gene for an enzyme called EPSPS that's involved in producing 3 amino acids in plants, because plants have to make their amino acids, we don't. And the way the resistance works is that a form of EPSPS that's much less sensitive than the plant's native form of EPSPS has been introduced from agrobacterium. They found one in agrobacterium that wasn't very sensitive. So what happens in the plant is when Roundup is applied, the version of EPSPS that's made by the plant binds up with the glyphosate and can't produce amino acids. But and that would have killed a soybean plant or a corn plant or a cotton plant. But in the case of Roundup Ready crop, there's also this other form that's still in there that came from agrobacterium, and it continues to produce the amino acid so that the plant's resistant.
Okay.
They're they're helping the natural environment because they greatly reduce the use of pesticides. In many areas of the world, Roundup Ready crops in particular have displaced the use of pesticides that are 3 to 16 times more persistent than Roundup is. And essentially what they've done is they've taken herbicides that only professionals can use, and we've replaced them with a herbicide that has such relative safety that will let people buy it in the hardware store or in the grocery store and take it home and put it in their gardens. So the main thing is greatly reduced the persistence and environmental risks of pesticides by replacing them with a pesticide that's pretty benign.
Okay. Does herbicide resistance affect any phenotypes of the crop?
No, that's one of the things that's been quite interesting. And again, it comes back to this business about EPSPS. There are hundreds of forms of EPSPS. Every plant makes at least one. Agrobacterium has some. Essentially what this shows, the fact that this herbicide resistance works so well, says that the form that's been introduced from the agrobacterium is so similar to the plant's own form that it's able to produce these amino acids still. Otherwise this wouldn't work. And if there were really adverse effects, it would probably mean that the crops wouldn't yield as well, but they do.
Okay. Do you have any opinions about how GM crops and herbicide resistance are changing the world?
Well, I think that it changed the world for the better. I hesitate, really, we don't want herbicide resistance to be the only way that GM helps improve weed management. And a lot of us are turning to focus on what we think is a longer term, more sustainable way of managing weeds, which is to improve the competitiveness of the crop against weeds. So we want to introduce traits like more rapid growth, resistance to lower dependence on nitrogen, more drought tolerance, and so forth. So that the crop has an advantage over the weeds and is able to outcompete it more.